Hi APE students, welcome to the soil testing pre-lab video. Let's get ready to talk about soil. You can tell as you read through the background information in your lab that the purpose of testing the soil sample that you brought in is to test two physical, we're actually testing four, not three chemical properties, and we'll use all of that data that we collect to determine if our soil is appropriate for growing alfalfa. In the next unit that we do on food resources, you will actually figure out a way to amend your soil and improve its ability to grow alfalfa, plant some alfalfa in both your non-amended and amended soil to see if you were able to adequately grow alfalfa. So let's go ahead and get started with the two physical properties that we're going to be assessing. The first one of those is called porosity, which is really the water holding capacity of the soil. And you can see from the simple diagram here that I'm showing you what pore space is. When we measure the porosity of our soil sample, we're measuring the amount of space, pore space, between the particles of the soil that can hold water and actually oxygen for cellular respiration in my plant roots. So the procedure for setting it up is pretty straightforward. The directions are very clear. You have a plastic tube that is open on both ends. You will mark that, fill it with the soil sample, take a small 2-layer piece of cheesecloth and affix that to the bottom and rubber band it to keep it in place. And you can see in the photograph below here what that sample looks like when you've got it all set up. You also have a plastic vial that your tube fits inside of. Once you've filled it with the soil samples, you then add a certain volume of water. Make sure that you're not adding the water to the plastic tube that has the soil. The water goes in the bottom of the vial. And what you're going to do, the theory for how this test procedure works, is that if you mass the dry soil column and you let it sit in water, capillary action is going to allow that water to move upwards through the soil. And if you mass your wet soil sample on the following day, you will be able to determine the water holding capacity. And I've also included for you a sample data table that you can take a look at in order to do those calculations. Notice that you're not just testing your soil sample, but you're going to compare it to a sample of pure clay, which has theoretically a very high volume of pore space, a sample of sand, and a sample of organic matter or humus. Those other samples, the clay, sand, and humus, are available for you. And the reason we want to do that is that knowing the water holding capacity of our soil as a standalone value isn't going to help us very much unless we can compare it to some known samples. The next physical property test you're going to do is texture. This is the relative amounts of sand, which, as you can see from the diagram here, is the largest of the measurable soil textures that we look at. So we will be measuring the relative amount of sand, silt, and the amount of clay, very tiny particles. The technique for doing this is to take this little urinalysis cup that will be provided for you, and you are going to do just your unknown sample. I will do a sample of clay and a sample of sand for you to compare them to. So you follow the directions in your packet where you're going to add some soil, your collected sample, which is the unknown in the diagram. You're going to add some water, you're going to shake this up, and you're going to let it sit overnight until the second day. It helps on that second day if you take the light from your cell phone and we'll kind of darken the room, and if you hold the light up to it, you're going to easily be able to see these layers. The particles, different size particles in your sample will have settled. And so as I look at my key here, obviously sand, the biggest particle, the greatest density particle, is going to form the layer on the bottom, followed by a layer of silt. And then again, as my key indicates, those tiny clay particles will form a layer on the top. And finally, any fluffy organic matter or humus that you have will be that very top, almost kind of floating layer. What I want to do with this on the second day then, and you can actually mark those different layers with a marker on your little urinalysis cup, and I want to calculate the percent of each of those different soil textures. So very simply, if I'm going to use some nice even numbers, if this bottom layer of sand, I'm going to measure the height of my entire column, and if the bottom layer of sand was, for example, 2 centimeters out of an entire column height of 10 centimeters, then I would say that my sample is a 20% sand texture. So I'm going to get the percentages of sand, the percent of silt, and the percent of clay by doing that same technique, measuring the height of the individual soil particle relative to the height of the entire column. And then I'm going to take those percentages and use my soil triangle and actually report my soil texture as a soil texture class. For example, maybe those percentages fall such that it is identified as a clay loam. You can see on your in your lab packet that you also have an extra credit option of doing a texture by feel test, which is the texture by feel is kind of like an if-then statement. So you start by taking some of your soil and getting it wet and squeezing it in your hand, and if it rolls into a ball, then you go on to the next step. The greater the amount of clay, the greater the stickiness of your soil, or it will stick together better. The chemical tests that we're going to be performing are all done with a Lamont soil test kit. Each lab group has the entire test kit at your lab table. And inside the test kit are all of the things that you'll need for testing the relative amount of nitrogen, phosphorus, potassium, and pH. Working in your lab groups of 4, on the first day, one pair should start the porosity test and one pair start the texture test. Those tests are finished or read on the second day, and the rest of that second day you should be able to each get 2 of the chemical tests done. You want to make sure that you have read the procedure for those, because some of them need to sit for a while before you can read them, and so you'll want to start those before the others, and while that one test is running you can maybe do another one. All of the equipment that you need is in the kit. So you've got little soil testing tubes with lids or caps on them, because you'll need to shake those up. There is a little brush in there for cleaning all of those. There are 2 different size scoopers. And again, the only tricky thing about the chemical test is reading the instructions carefully. One of the little scoopers is a 0.5 gram and one is a 0.25 gram. So you just need to make sure that you're getting the right quantities of soil. All of the other reagents that you need are in clearly labeled bottles, and you have an envelope that contains color test charts for you to record your final answer.